So I'm going to get going now, actually, and just see if people trickle in. So it's nice to meet you all. Thanks for coming. I'm Oscar, one of the fifth years. I'm going to be lecturing tonight, just going through a revision of somatosensory systems, which is quite a lot. So I'm just going to cover it fairly superficially. And then if you have any particular questions, my email's on that slide, and I'll put it at the end as well. So do feel free either to ask today or to email me and get in touch. So just broadly, what we're going to cover, what actually is somatosensation, what are the receptors involved, and then there's two main systems that you will be aware of, the dorsal column system and the anterolateral system. And they differ in lots of ways, but you know, there's key sort of fundamental differences between them, and that'll help you remember the key characteristics. We're going to talk about some clinically relevant spinal cord lesions, both because they're likely to come up on your exam, but also they really help you understand the neuroanatomy, which probably will be tested as well on your exam. Finally, obviously your brain's involved in somatosensation. So we're going to sort of work up from the receptor to the brain, stopping off at the important bits along the way. If I go too fast with anything, obviously just feel free to put it in the chat or to unmute. So starting off with what actually is somatosensation. So it's basically just your body's ability to perceive and interpret any physical sense. It doesn't have to be outside the body, but it mainly is. And some of it is specified towards noxious stimuli, so anything that manifests as pain. And that can be painful heat, painful chemicals, painful touch. But then you also have specialized sort of temperature sensation, chemical sensation, proprioception, which is your ability to sort of detect where your body is in space. Then you have crude touch and fine touch. And importantly, all of these different modalities of somatosensation are carried by different, so they're transduced by different receptors, and then they're often split into different systems based on how they get up the spinal cord. So the neuroanatomy is really illustrative. So thinking about any receptor, receptors vary in lots of different ways. The first, as we've discussed just then, is that they vary in what they actually respond to. So for example, some receptors are adapted to temperature and only really detect temperature. But within each modality, you've also got variation. So the sensitivity of receptors, whether they adapt or not, and how quickly they adapt. Adaptation is really important. We're going to talk about it using the Pacinian corpuscle as an example, just because it allows us to detect things in a more dynamic way, and therefore draws more attention to things that are probably more relevant in terms of new stimuli or stimuli that are changing. And as I've been sort of alluding to, the fibers that carry these impulses from different receptors differ, and that then feeds into different systems. So as I touched on, we're going to focus on the dorsal column system and the anterolateral system. So just briefly, because we've sort of covered this, but just to be aware that there's lots of different receptor modalities. So mechanical forces are transduced by mechanoreceptors, heat or cold thermoreceptors, chemical changes, chemoreceptors, and then you have specialized nociceptors as well, which can be sort of any of them, and they detect noxious stimuli. So anything that's sort of evolutionarily telling you to get away from. And as I said, you've got different nerve fibers, which have very different characteristics. And importantly, the different nerve fibers, which vary both in whether they're myelinated and their diameter, carry impulses from different receptors. So as a broad rule, you have large and small fibers and myelinated and not myelinated fibers. And the more myelinated, larger fibers tend to carry impulses much faster. And you'll have covered that in your sort of basic neuroscience why, in terms of like the length constant and the time constant and things like that. and they tend to carry impulses from your proprioceptors. So things like your reflexes have to be really fast, and so that makes sense evolutionarily. Whereas your small fibers, which tend to not be myelinated, and the name suggests are very small, are quite slow, and they tend to carry pain and temperature. So I'd be aware of this table. It's something that's often tested, but it's more that the principles of it, as opposed to, you know, particularly specific speeds and things like that. So just using the Pacinian corpuscle as an example, it's a type 2 mechanoreceptor, which that's just shows that mechanoreceptors can be classified into different types. But really importantly, as an example, it's rapidly adapting. And as I said before, that ensures that it responds only to sort of new or changing stimuli, as opposed to if someone's just applying a constant pressure, it's going to stop firing. And that's important because that's not useful information. You know that there's a stimulus there. And as I said before, it is associated with A beta fibers. So looking back at them, these are thickly myelinated, they're medium diameter, and they have quite a decent speed. So that would still be classified probably more as a larger fiber. Likewise, not only are they rapidly adapting, but they're also relatively selective for vibration rather than just static compression. So in, for example, an essay, if you were asked to talk about like receptor adaptations, the Pacinian corpuscle has been done to death, but it is important as an example. It's the one I've chosen today just to illustrate how receptors differ in their characteristics. We definitely don't have time to just go through like a laundry list of receptors. I won't subject you to that. But there should be more information about the different types in the notes that accompany this revision lecture. So in your syllabus, it mentions being aware of how force transduction works. So as you know, neurons work by gated ion channels, allowing depolarization. Once the impulse is going, it's voltage gated ion channels that perpetuate that impulse. But to get the impulse started in a sort of somatosensor, it depends on the modality. So this is a mechanoreceptor. So what it's doing is converting physical energy into electrical energy. So that physical energy comes in the form of, in the Pacinian corpuscle, as an example, typically vibration. So if you imagine each vibration as deforming the membrane of the corpuscle itself, it transmits that mechanical force to the nerve fiber ending inside it. That deformation then allows sort of stretch gated sodium channels to open, starting the impulse. So that's just one example, but be aware that different types of somatosensor obviously transduce different forms of energy or different stimuli. So as an example, a chemoreceptor might respond via like direct pores and things like that. We won't go into that. So a bit of experimental evidence just on the Pacinian corpuscle, some very famous experiments by Lowenstein in the 60s. So the first sort of set of experiments demonstrated that it's very adaptive to mechanical stimuli. And as we've said quite a lot, that just means it eventually stops responding. So what they basically did was compare the complete Pacinian corpuscle with one where they'd stripped the outside capsule from. And they showed that when they stripped the capsule away, the naked nerve sort of continued to respond to a steady pressure, whereas the complete Pacinian corpuscle adapts quite quickly. So they suggested that partially it's the capsule itself that mediates this adaptation. There probably are other reasons why it adapts. So, for example, the nerve itself might adapt as well. The second set of experiments, they showed that it's relatively selective for vibration, as we mentioned. So it's not just steady compression. So as we said, a mechanoreceptor has to transfer sort of a physical force to actually deform the nerve. And how it does that can sort of control the properties with which it responds. So the proscenium corpuscle, when it's being steadily compressed, acts quite well to absorb that pressure. So it doesn't really deform the nerve much. Whereas if it's being dynamically compressed, i.e. by something vibrating, they basically showed in these experiments that the actual viscosity of the proscenium corpuscle itself increased. So the force from the outside was transferred better to the nerve. So in short, the more dynamic the stimulus, the more likely it is to respond. And clinically, the modality of nerve fiber is really important. So the first example, which is very commonly touched upon, is local anesthesia. So quite a helpful fact is that local anesthetics such as lidocaine, which block voltage-gated sodium channels, are really selective for the smallest fibers, which fortunately carry pain. So first of all, that leads to preferential blockade of pain, where there's still preservation of touch. So if someone's under local, you might expect that whilst they won't be able to feel pain, they'll still have a sensation that, say they're having an operation, some orthopedics things are done under local as an example. They'll still know that something's happening and they'll still feel like a dull touch, but they won't feel the pain, fortunately. The other is that this blockade is use dependent. So, you probably know this well, but I'll just quickly go over it, that the local anesthetic has to get into the inside of the nerve because it blocks the inside of the channel. And so the more quickly the nerve is trying to fire, the more open those channels are, and so the more it gets blocked, which again is useful and fortunate clinically, because it means those fibers that are getting aggravated the most by a stimulus are more likely to get blocked. The second sort of really clear clinical example of these different types of nerve fiber is that they can be tested neurophysiologically, and it gives a really good picture of what's going on in, for example, a neurological presentation. So there's an entire specialty devoted to doing these tests called neurophysiology, and they work really closely with neurology. One of the things they do is nerve conduction studies, and importantly, they can demonstrate based on the speed of transmission that's affected, i.e. whether it's large fibers or small fibers, what might be going on. And they can also show whether the nerve fibers affected are being demyelinated, i.e. the velocity of the impulse is decreasing, or whether they're actually losing their axon itself, where instead of having a slower impulse, you get a lower amplitude impulse. And as we've discussed, there are two really key systems in somatosensation. So the first is the dorsal columns, and then the medial lemniscal system. So that's all one system, which mainly carries fine touch, vibration, and proprioception. That's really adapted to provide accurate, detailed information, and we'll discuss how that happens. Whereas the anterolateral system, which is sort of its main comparator, instead of carrying the more fine detail, carries pain, temperature, and crude touch, which are more sort of primitive. They're more likely to be noxious. So this system's really adapted to provide quite basic life-preserving responses, but less detail as to where exactly they're coming from. So here's your spinal cord in transection. This is the cervical spinal cord, so it's got information from both the legs and the arms. And the main stuff we're going to talk about today is up at the top, the posterior or the dorsal columns. and then down at the bottom in blue, the anterolateral system, which here only has the spinothalamic system, but you guys have to be aware of a few other components of that. For example, the sides here, the spinocerebellar system. So we're going to cover all of them in brief, and then if you have any questions, you can ask. So just the anatomy to recap of the dorsal column system. So the first order neurons, which is the actual sensory neuron itself, doesn't decussate, and it doesn't form a synapse until it gets to the medulla. So it goes straight into the spinal cord, ascends on the same side, which is really important, and it goes to the dorsal column nuclei. As it goes up the spinal cord, the legs and the arms are split. So the legs are in the middle in the gracile fasciculus, and the arms are more laterally in the cuneate fasciculus, and that corresponds to the nucleus of the medulla that they go to. So as you can see on this diagram, I know it's very small, but I wanted to keep it quite simple. The lower limb is medial, and that goes up to the nucleus gracilis, whereas the upper limb is more lateral, and that goes up to the nucleus cuneatus. So just to be aware of the separation of upper and lower limb and dorsal columns. So that's still your first neuron, and then we get to the medulla, where it synapses to form the second order neuron. The second order neuron immediately decussates. It then forms something called the medial lemniscus, which is, for the vast majority of its course, is now contralateral to where the stimulus originated, and therefore on the same side of the brain as where this information is going to be represented. So it's really important to recognize that this decussation, which is really important in neuroanatomy because it decides whether things are ipsi or contralateral, really important to recognize that this happens very high up compared to the other system we're going to talk about. So this happens after the medulla, very shortly after, essentially in the medulla. And so, the medial lemniscus takes us up to the thalamus, which is essentially the main relay in your brain, whether motor or sensory. Lots of things go to and from the thalamus, so it's no surprise that this system goes there. Finally, from the thalamus, you get the third order neurons, which is shown in blue here. They ascend in the posterior limb of the internal capsule, and they go to the somatosensory cortex, which, as we will discuss, is where all of your sort of somatosensory information is kept. So, as I said before, the dorsal column system is really adapted to providing quite intricate information. It preserves the sort of somatotopy, which is basically where exactly on your body the stimulus originated, and it preserves that all the way up to the cortex. And so you get a really good picture of where exactly the stimulus originated. And so the dorsal columns are really important in things like 2-point discrimination. And there's two sort of sensory coding features which are alluded to in your syllabus, which maintain this sort of really accurate and precise representation of information. So the first is that the signaling is maintained in parallel. The other option would be for signals to converge, like they do in the spinothalamic system. If they were to converge, it means it's probably more sensitive and more likely to fire, but then you wouldn't know where exactly the most stimulus was coming from because it's all just blending into one. In contrast, here in the dorsal column system, whilst it's sometimes less sensitive and so less adapted to getting you out, quickly from, a hot stimulus or something like that that's going to burn you, it allows you to know exactly where, for example, 2 points are on your skin, for example. Another related concept is the concept of surround inhibition. So this is basically where in the medulla, in the dorsal column nuclei, which is where the third neuron meets the second order neuron, lots of inhibitory interneurons create a field of surround inhibition. So if one sort of region of neurons is firing really strongly, not only will they be firing more strongly than the neurons around them from the start, because they're sort of being stimulated by their receptor, they actually actively inhibit the surrounding area. So that massively increases the potential to distinguish between 2 points, because between them, it's almost like your dorsal column system is actively turning off a stimulus. It's even better than there being no stimulus, if that makes sense, because they're actively enhancing the contrast between them. And the first experiments to show this made some really good graphs, which sort of show this in principle, where a stimulus looks like a mountain, and when you have no surround inhibition, between the mountain there's sort of still a bit of a hill, whereas with surround inhibition you get a mountain, then a really clear trough, then another mountain. I should have just got a picture on that rather than describing it, but it's something to look at if you struggle to understand the sort of principle of it, and that was probably when I first understood it. Another important feature of the dorsal column system is that it's subject to a lot of corticofugal control. So corticofugal just means coming back from the cortex, so coming sort of from brain downwards. So neurons of the cortex are adapted in the dorsal column system to provide lots of feedback back down to the lower neurons, for example, in the medulla or further down. And that allows for something called egocentric selection, enhancing the sort of representation of stimuli that are thought to be quite important. So you can sort of conceptualize it as if someone was asking you to distinguish 2 points on your skin that they were touching simultaneously or something, and asking you, is it sort of 2 points or one, or if they were drawing a letter on your skin and asking you what was being drawn. If you were really focusing on it, this might be one of the systems that helps you to enhance your ability to detect that. So just going back to this picture of the spinal cord, so we've covered the posterior columns now here, and just to recap, they carry fine touch, proprioception, vibration, things like that, whereas your anterolateral system carries more sort of primitive things like pain and unconscious responses, temperature, and crude touch. So we're going to discuss them now. One feature, I don't know quite how detailed your sort of neuroanatomy questions get, but one thing to remember with the dorsal column system, as we said, is that the legs are inside the arms, and that's actually reversed here. It might be slightly difficult to see, but for example, in the spinothalamic system, I know you probably can't see my cursor, and that's probably quite annoying for you guys, but you can see that the arm is more medial than the leg, so it's just the inverse. So if you remember one, just remember that they are opposite to each other. So now we're talking about the anterolateral system, which is sort of the opposite of the dorsal column system in a way. First thing to be aware of in the anatomy, which we're going to cover in a similar way to the dorsal columns, is that there's a thing called Lissauer's tract. And so the first neuron, i.e. the neuron that connects to the receptor, the mechanoreceptor, for example, or the nociceptor, often sort of gets into the spinal cord but messes around a bit and either goes up for a few levels or down for a few levels before actually going into the grey matter and making a synapse. So it's just something to be aware of, and it does actually have some clinical relevance at times, and I'll show you why later. So Yeah, so on this picture, you can't see Lassalle's tract because just imagine that it's already done that. So at the bottom, this red neuron would not necessarily go straight into this grey matter. It might go up or down a few levels. But imagine it's done that. So it's done its business in Lassalle's tract. In contrast to the dorsal column system, it immediately synapses. So straight away in the dorsal horn, the grey matter, it immediately synapses and forms the second order neuron, which then, so one area which is really important in terms of the first order neuron synapsing is the substantia gelatinosa. And again, we'll talk about why that's important and some examples of it. So here it is. This is just the grey matter of the spinal cord. So you've got to imagine a circle around this with all the white matter tracts and things like that. So this is more the area of like processing and synapses and things. You might be aware that the spinal cord grey matter is arranged in laminae, which is just layers. Laminae 1 to 4 are the dorsal ones in the dorsal horn. They're the most dorsal, and they're the major sites of synapses of the anterolateral system, particularly the spinothalamic. And the laminae 1, the substantia gelatinosa, it might be, don't quote me on that because it might be a few levels. It is a key site of pain processing, which we'll cover shortly. So as I said, the second order neurons decussate really soon. So if you remember with the other system that we're talking about today, the dorsal columns, it's the first order neuron in that case, but it rises throughout the entire spinal cord on the same side as the stimulus. Whereas here, whilst it's still the second order neuron decussating, the second order neuron has formed much sooner. So contrast that within the medulla for the dorsal column system. The second order neuron here is forming only, potentially a few spinal levels higher, but it's still in the spine. And then it immediately decussates, which means for the vast majority of your spinal cord, the anterolateral system represents information from the contralateral body having decussated. This decussation occurs in the anterior commissure, which has clinical relevance in something called syringomyelia, which we'll discuss later alongside another classical picture of a spinal cord lesion called Brown-Séquard syndrome. So just to summarize, key points here are that, first of all, the first order neuron can go up or down a few levels in the Lassalle's tract. Second of all, it synapses to form the second order neuron, only a few spinal levels above or below the initial stimulus, whereas in the dorsal column system, that would be up in the medulla. So, and therefore, because it decussates early, it carries information on the opposite side to where the stimulus originated for most of its way up to the brain. So that second order neuron that comes up the spinal cord contralaterally gets to the thalamus eventually. As I said before, a key relay in all of neuroscience, so it's no surprise, and it's a different, very slightly different bit to the dorsal column system. There in the thalamus, it forms third order neurons, which then ascend to the somatosensory cortex, again via the internal capsule. So this bit is quite similar to the dorsal column system once they've got to the thalamus. So the anterolateral system isn't just the spinothalamic projection. It's definitely the most important and sort of one you will study the most, but it's just important to remember that there are a few others. So just to recap, the spinothalamic projection goes to the ventroposterolateral or VPL thalamus, and that's involved in your conscious perception of pain, temperature, crude touch, and firm pressure. It's split, if you're interested, into two. So the anterior part carries crude touch, whereas the lateral part carries pain and temperature. But it's not just that. So the spinomesencephalic carries information up to the midbrain, specifically the periaqueductal gray. And the periaqueductal gray, you might remember, is a really key sort of control center of pain. So, for example, if you stimulate that region, you can basically eliminate your perception of pain. So the thought is that your spinomesencephalic projection basically activates descending control of pain, which we'll talk about very shortly. Finally, your spino-reticular system. That's main target is your reticular activating system, which is really important in your consciousness, in your level of awareness and alertness. So again, just showing that the anterolateral system has specialized subsections, and they're really important in different things. So as I said before, partially controlled by the spinomesencephalic projection up to the brain is the fact that the anterolateral system, as one of your as your main carrier of pain, is subject to modulation, and that's really helpful evolutionarily, because whilst it's important to perceive pain and to know that there's a painful stimulus, if you're in too much pain, you might not be able to get on with the sort of life-saving intervention. And so your brain has some quite profound ways of turning off pain signals. So one might be, so both of these occur really in the substantia gelatinosa, which we discussed before, and it's just highlighted on the bottom of this slide. And there's two main ways this occurs. So the first is endogenous opioids. So endorphins, enkephalins, and dynorphins are all examples of these. You might know endorphins from sort of going on a really long run or something like that. So essentially that they act in this region, they cause potassium influx, and that hyperpolarizes the neuron, preventing it from firing. The second key process involved in modulation of the anterolateral system is called gating. So gate theory was sort of first formally conceptualized by Melzack and Wall. And there's neuroanatomical evidence for this. It does happen that essentially in the substantia gelatinosa, there are projection neurons which ascend up in the spinothalamic tract, and they might be targeted by both the nociceptive fibers shown here in red, but also by larger fibers, for example, an A alpha or an A beta axon, which might be from like a mechanoreceptor, so something that would be triggered just by rubbing your arm or something. And between them is an inhibitory interneuron. Really importantly, the larger fiber, the mechanoreceptor, can act to activate this inhibitory interneuron, essentially turning off any signal from the nociceptive fiber to the projection neuron. And so these sort of dorsal column neurons can essentially modulate your experience of pain. And so this is at least some of the neuroanatomical basis for why rubbing it better can really work. So larger mechanoreceptor fibers can effectively turn off the signal from some smaller nociceptor fibers. Finally, just something that's mentioned, I've seen in your syllabus, is the spinocerebellar tract. So that carries proprioceptive information to your cerebellum, but importantly, it's sort of your unconscious processes and your coordination. So you're not really aware of this, but it's constantly working to keep you upright and keep you coordinated. So it's highlighted on the right. It's sort of the most lateral in the spinal cord, and it's again got an anterior and posterior, but I wouldn't worry about that at all. So the sort of final major section of this is we'll also talk about the brain, but we're going to talk about how lesions manifest in the spinal cord. So hopefully from the neuroanatomy we've revised, you'll be able to tell me, or at least we'll go through together some of these examples of spinal cord lesions. So if you're presented with a question about a spinal cord lesion, the sort of basic approach can be to characterize where the lesion is. First, to be aware of the reflexes. So biceps reflex C5, triceps C7, knee L2, 3 and 4, the ankle S1. And if they're still functional, it gives you some idea of the preservation of at least that bit of spinal cord. Next, vertical information. So the arms are controlled by C5, 6, 7, T8 and T1. So if the arms are involved, you know that it must be at or above T1. A T5 lesion isn't going to affect your arms. If the diaphragm's involved, the diaphragm's controlled by C3, 4 and 5. So again, it has to be at least above C5. And finally, you can identify the sort of nature or side of the lesion based on the modalities affected. And this is really a sensory lecture, but obviously the spinal cord also carries motor information. And so you can work out based on the pattern of weakness as well. Typically that'll be on the same side as the dorsal column for that bit of body. So let's go through some examples. So the first is syringomyelia, which occurs when, if you look at this picture of the spinal cord, you'll see a grey circle in the middle. And that's just trying to show what's called a syrinx, which is basically just a fluid-filled cyst which can form in your spinal cord from things like tumours, from trauma, but it can also just occur on its own, so idiopathically. And so this syrinx expands, and so it affects the fibres nearest to it. And the fibres that are most affected, or that are affected soonest, so often the fibres that are affected when someone presents with the features of syringomyelia are the anterior commissure. So if we think back to the neuroanatomy of the anterolateral system, you'll remember that the second order neuron, having formed, having begun in the dorsal horn of the spinal cord, then quite soon after decussates in the anterior commissure to then ascend on the opposite side of the body. And it's in this decussation that the fibers are affected. So remember that in this anterior commissure, fibers are decussating both ways. So on this diagram, you can see from the patient's left, the fibers decussating to their right, but remember that obviously fibers will be coming from their right and going to their left. And so you get bilateral bilateral fibers. The fibers only at this level are affected, so only at this sort of associated levels, because it's only the fibers that are decussating. If the fibers already have decussated, whichever side they're on, or if they're not going to decussate soon, then they're not affected. So it's only the fibers that decussate around the level of the syrinx that are affected by the syrinx. It's just mechanical compression of it. So you get what's called a cape-like distribution, sort of as shown on this picture. But if you look to their back, it would sort of go down their back as well, just by virtue of how your dermatomes work. Importantly, because this is only the anterolateral system, you get a loss of pain and temperature and crude touch, but you still have preservation of things like fine touch and proprioception. So a classic clinical vignette would be, for example, someone has found that they're just repeatedly burning their hands, but they can't feel it. And that shows two things: they can't feel pain or temperature, and that the hands are affected. So immediately should be thinking syringomyelia. The next is two examples which affect the dorsal columns in particular. The first is subacute combined degeneration. So as the name suggests, that's not just the dorsal columns, that's also the corticospinal tract, which you'll have covered in your motor neuroanatomy. So in that you get bilateral loss of your dorsal column function, so proprioception, fine touch, things like that, vibration, as well as bilateral weakness. That results from a deficiency of vitamin B12. So there can be lots of reasons for that. Interestingly, nitrous oxide, so NOS, if you do loads of NOS over a short period of weeks, it really messes with your B12 metabolism. And so you can actually precipitate subacute combined degeneration from NOS. Another example is tabes dorsalis. I've definitely said that wrong, which essentially results in what occurs in neurosyphilis, and you get quite specific involvement of your dorsal columns. So you'll learn later clinically what someone would present like when they don't have their dorsal column function, i.e. what the consequences are of having very little or no proprioceptive ability, fine touch ability, and sensation to vibration. But just be aware that these are sort of classical cases which knock out specifically the dorsal columns and or added features like in subacute combined degeneration. So moving up to the brain, your somatosensory cortex is in the postcentral gyrus. You'll all know that. You'll probably also know that it's arranged somatotopically. So the way I always like to remember it is as if a little figurine, as shown in this picture, is sort of draped over the brain, sort of hanging by their ankles. And so you remember that the feet are in the midline, whereas the head is sort of dangling backwards with the hands. So from medial to lateral, you move from your legs to your face. But as shown in this picture, you've got what's called the sensory homunculus. And as you'll note, they don't look quite like we do, because parts of their body are massively overrepresented. This overrepresentation is covered more in the notes I've written for this, but essentially it's just directly proportionate to the density of receptive fields. Because in the brain, each receptive field has sort of a similar size, yet in your sort of more peripheral tissues, you might have way more receptive fields per square inch in your hands than, for example, your abdomen or something like that. So to represent the total area of the hands, you need much more brain space. And obviously that's functionally significant because the regions that do have this overrepresentation are necessary for this function. And clinically, this is relevant in strokes in particular. So if you just think back to the previous slide of your sensory homunculus hanging down by their feet from the brain, you'll know that the anterior cerebral artery in yellow supplies the midline. So this is a lateral view of the brain, but if you saw it medially, it'd look much more yellow because lots of the medial surface is supplied by the ACA, whereas the middle cerebral artery in red here supplies more the lateral surface of the cortex. And so just to recap, I'm sure you will know this well. If you imagine the figure sort of hanging by their legs, anterior cerebral artery infarction is going to affect the leg mainly, whereas middle cerebral artery is going to affect the face and arms preferentially. They're not mutually exclusive, and a lot of strokes you get crossover and you get features of both, because both arteries have been blocked. So just one practice question. If anyone would be keen to go through this, feel free to put it in the chat, or if you're feeling like you'd rather unmute, please do that. So this is a patient who has had an injury and had straight away had a pattern of sensory loss and motor weakness. So in this sort of black bar here, number 1, they've lost all sensation in that region, and they're paralyzed. In this orange region in number 2, they're also paralyzed, and they've lost their vibration sense and their proprioceptive sense, as well as fine touch sense. In 3 in green, they've lost their pain and temperature sensation. So first of all, if you could put in the chat, if you're willing to, what kind of lesion would cause this, and where it would be, whether it's on their right or their left, and which fibers have been affected of the spinal cord, not which fibers, but which tracts. And then if anyone knows, so there's a bit of an asymmetry here where the loss of the pain and temperature in green is a bit lower down than the loss of vibration, proprioception, and fine touch on the right. And there is a reason for that. There's probably several reasons for it, but if someone knows that as well, I'll just give you a few minutes to think through that. Yeah, great. Thank you, Grace. So that's exactly right. So, this is something that will be probably featured on your exam somewhere, because it's just so illustrative. It's not a common lesion to actually get clinically, because it has to be from something like a stab wound or something that somehow takes out half of the spinal cord. But it's really illustrative of the anatomy. So exactly, it's on the right-hand side. So yeah, a hemilesion on the right, around the sort of level of the black band. If you remember that the black band, they had sort of total loss of sensation and sort of total paralysis. That's because both the upper motor neuron and the lower motor neuron are affected there, and also all the sensory fibers on that side before they've had a chance to decussate or sort of enter the ascending tracts have been affected. And then exactly as you say, because it's the right-hand side, you're going to get the loss of the dorsal column function on the same side and loss of the anterolateral column function on the opposite side. And exactly as you said, because the fibers that eventually form the anterolateral system might go up or down a few levels in Lissauer's tract. That means that they might escape this lesion. So it's not something to dwell upon loads, but just remembering that can explain why you get slightly weird pictures. So this would be the lesion that's caused this clinical picture. If you just imagine that the neurons representing the little white square sort of at the level on the other side that still has sensory function, would still be on this side, so they haven't yet entered here and decussated. So I mean that's the end of what I wanted to cover today, because I had to keep it fairly I couldn't go into too much detail about anything, because the actual section for this in your syllabus is huge. But I wanted to just go through the two main systems, how they differ and why that's important. And so if you have any questions, feel free to put them on the chat now. Feel free to email me. I'm just, for anyone that joined late, I'm just going to post the feedback link again, which if you fill that in, I think you then get access to the notes I've made that accompany the lecture.